Okay, good morning everyone. So we're going to start. Welcome to the seminar series. So as usual, we are on webinar mode. So people online, do not hesitate to report technical issue in the chat and to ask your question in the Q&A tab, and we will allow you to interact with the speaker at the end of the seminar today. And with that, I thank very much, Luis, for introducing today's speaker.
So hello everyone, it's a pleasure to have today Dan Weinreich as a speaker. So before I forget, Dan is visitor in sabbatical for one more month, I think. So feel free to contact him and have a chat. He's staying at my office here at the set. So even though he was originally invited by Nacho, saying hi to him if he's online. He moved on to another sabbatical, Nacho. So Dan was originally a computer scientist and he was an engineer in computer science for 10 years. And then he decided to pursue, to do a PhD in evolution, and he decided to basically start a PhD with Leontine. Who better than him to train him on that? After that, he did a postdoc doing experimental work that people might not know with Lin Chao. And he followed up with a postdoc with Dan Hartwell at Harvard for a few years. And since 2007, he's been professor at Brown, so where he's till now. So the floor is yours.
Okay. Thank you, Luis. For the introduction, thank you all for coming. As Luis said, my name is Dan Weinreich. I'm a professor at Brown University in Providence, Rhode Island. This is, in fact, my second sabbatical in Montpellier. My first was in 2021. You may remember the pandemic. And so my wife and I had a very interesting and very enjoyable 6 months here, but that did not include coming to campus more than about 3 times with social distancing. So it's been a great pleasure to spend the fall semester here in this building with Luis's team and getting to know other people at the staff. And as Luis said, I will be here until the middle of January, and our ambitions are to come back for a month or 2 next summer as well. So I would be more than happy to sit down and chat with anyone for whom this work is interesting or who have other topics they think maybe would be fun to discuss. Okay, so I will present a lecture now on recent work in my group with a couple of collaborators, including Nacho Bravo. on the population genetics of biological noise. Let's see, will this work? Yeah. Up and down. Good. Okay. So to begin at the beginning, information transmission is intrinsic to biology. Each generation, newborn organisms develop into adults using information that they are born with. And then during reproduction, the adults transmit some information back to the newborn. This kind of bidirectional information transmission was pointed out by John Maynard Smith. Noise is intrinsic to information transmission is intrinsic to biology. Noise is intrinsic to information transmission. So the information of the newborn inherits from the parent is in one way or another sure to be corrupted during the course of development. And similarly, the information that the adult intends to transmit to the newborn will again be in part corrupted during the process of reproduction. Noise enters during development, for example, as a consequence of stochastic variation in the amount of mRNA in a cell, as a consequence of transcription and translation error, as a consequence of proteins not folding correctly, and so on. But critically, for our purposes, while developmental noise is random, its intensity can be influenced by the genetics of the organism. So I'm showing you some results from a paper by Olin Sealander when he was a postdoc with Myron Ackerman at the ATH. And what Olin did was to take 1,500 different promoters of the E. coli genome and attach each of them to a green fluorescent protein gene. And then by watching populations of cells carrying one of these constructs and recording the intensity of green fluorescent protein expression or fluorescence in an ensemble of cells, he was able to get a sense of the variation in expression level due to each of these 1,500 different promoter genes, alleles. And so here are some representative data. You can see that for these 5 promoters, there's quite a range in mean expression, but there's also quite a range in the spread of these distributions. Okay, the x-axis is the intensity of expression and the y-axis is the density or the proportion of cells that are expressing at that level. Here are the data in total. So there are 1,500 points in this scatter plot corresponding to these 1,500 different promoters. The 5 at left are shown by red dots with labels. The point I want you to take is that while there is a wide range in mean expression level, there's also a wide range in standard deviation in expression. These two quantities are correlated. That's perhaps not surprising. But what I really want to focus on is, given any particular mean expression level, there are alleles that have higher and lower variances in expression. The genetics of the organism can control the noise in gene expression. Similarly, noise enters during reproduction. This is perhaps even easier to see, or at least has been recognized for longer. So this happens during mutation. In diploids, it happens during meiotic segregation and syngamy. In any organism that undergoes genetic recombination, that introduces noise. Here, what I mean is that the offspring don't look just like their parents due to these kinds of processes. More recently, we've become interested in microbiome and vertical transmission of microbiomes in organisms that have microbiomes, and there can be some noise in the precise composition of the microbiome that the offspring acquires from the parent. Again, though, while reproductive noise, not focusing on reproductive noise, although it's random, its intensity can again be influenced by genetics. So here are some data from the last century in showing this was a sample of sort of 50 E. coli isolates. Some are pathogenic on the right, others are commensal on the left. And the authors measured the mutation rate in these sort of 50 different isolates. You can see the y-axis is log transformed. So there's quite a wide range of mutation rates. Okay. So biological noise is random, but its intensity can be genetically influenced. And what I want to represent here is the kind of experiment that one would do in order to demonstrate this genetic control of biological noise. So here we're focusing on modifiers of development. And here here are a bunch of newborns, and on the left, and they're carrying a capital A allele, and that capital A allele has some influence on the developmental process, leading to a wide ensemble of adult phenotypes. On the right, we have another ensemble of organisms that are genetically identical, except they carry the little a allele at this locus, and the little a allele has some influence on the developmental process, but the net effect is that the variance, the variability in adult phenotypes is smaller. So we would call the allele, the capital A allele, a high noise allele, the lowercase allele, a low noise allele. This is the story for modifiers of development. Here is the story for modifiers of reproduction. The thought experiment, or the experiment is you take an individual in panel D, an individual carrying a big A allele. This is maybe a mutator. And so the offspring that are produced inherit more variation. On the right is an anti-mutator, a wild type allele, the little a allele. The same individual produces 5 offspring that are genetically more similar to each other and genetically more similar to the parent. Okay. And I should say that I've given you just 2 examples of these experiments, but since the turn of the current century and the advent of genomics and single cell microfluidic microscopy and so on, there are tremendous technical opportunities to precisely measure the variation in phenotypes, in the case of the upper, or in inherited in inheritance in the lower panel. Generalizing previous usage, I propose we call these alleles, the capital A and lowercase allele, modifier alleles. And if you're familiar with the modifier literature in the sense that's been used by Lee Altenberg and Marcus Feldman and others, this is not an accident. I would argue that the work I'm going to present today is a generalization of that earlier work, and I'll come back to that point at the end. Okay. So today I'll present a novel theory that describes the population genetics of these modifier alleles that heritably influence the amount of biological noise in any organism. This work came out of a graduate seminar that I led in the spring of 2020, entitled The Evolution of Evolvability, about a dozen graduate students, undergraduates, and other interested people. And I met every week to read a bunch of papers in this area. And then the work sort of lay fallow during the pandemic, but 3 people, Shenga Petak, a graduate student at the University of Vermont, Tom Skouros, a colleague of mine at Brown, and Nacho Bravo, who's known to most of you, came back together, and we've kind of hammered these ideas into a manuscript. Okay. So here's the outline for today's talk. I've already given you the problem statement. Under what circumstances will natural selection favor high noise alleles? Under what circumstances will favor low noise alleles? We'll begin by exploring a toy model that we've developed that explores, that predicts the fitness effects of alleles that influence biological noise. We'll then draw on work from others to think about how such an allele traverses through the population to fixation, and then implications for modifier theory. I've already alluded to that, and evolution of evolvability, which is actually, in my view, the exact same problem. And then, time permitting, I'll say a few words about what we're doing now. Okay. Fitness effects of biological noise. This is the very simplest model I could conceive of. This is a one-dimensional scalar phenotype represented by the letter Z on the x-axis. And I made up the simplest fitness function, this quadratic function that asserts that the fitness of any particular phenotype is given by that phenotype squared subtracted from some optimum. Okay. It turns out that almost trivially without loss of generality, I can make the optimum phenotype be at value Z equals 0. Not quite as obvious, but the value of the Malthusian fitness that I assign to that phenotype is also arbitrary. And the reason is because natural selection acts on differences in Malthusian fitness. And so if this curve is moved up or down, but otherwise not distorted, the consequences for evolution are changed. So just for illustrative purposes, I'd say it's 10. Here is some particular organism in the population. Its phenotype is Z0. Its fitness is R of Z0. In this case, Z0 is -2, which makes its fitness 10 minus -2 squared, or 6. But now let's allow some noise in the realized phenotype of this organism. So the Marigold distribution is a normal distribution with some variance, with some standard deviation sigma. And it turns out with some help from Mathematica, you can take that normal distribution and push it through this quadratic function and get out a new probability density function for fitnesses shown in blue. Okay. So here is now that probability distribution, the probability density function, given some value of Z0 and given some value of sigma, I can compute the probability of any particular fitness being realized by that organism. Okay. And phenotypes that are to the left of Z0's fitness, less than 6, are deleterious compared to the parental phenotype. And phenotypes to the right, phenotypes that have fitnesses greater than 6, are beneficial. Okay. Here is the exact same function, but I've increased sigma. So the blue line corresponds to a sigma of 0.5, and the gold or marigold line corresponds to sigma of 1. So it's more spread out. Increasing variance in phenotype, increasing standard deviation in phenotype, increases standard deviation in fitnesses. Two things I want to draw your attention to about these two functions. The first is if I focus only on the expected fitness across all noise. In other words, I have an organism, its phenotype is Z0, the offspring's phenotype is drawn from this normal distribution. Its fitness then emerges from this mapping. And I average over all noise, the fitness of the offspring goes down with sigma. So what that's illustrated here, the purple line to the right, the one that's closer to 6, corresponds to the expected fitness across all noise for the low noise distribution, the blue distribution. And the purple line to the right corresponds to the expected fitness across all noise for individuals that have the high noise allele. Noise drives expected fitness down. That's actually to be expected. It emerges from the concave down fitness function, so that we can now appeal to a mathematical theorem called Jensen's inequality, that increasing the noise on the x-axis reduces the mean, holding the mean constant on the x-axis, but increasing the variance drives down the mean on the y-axis because the function is concave down. Basically, excursions to the left and right are equally likely, but excursions to the left are more painful than excursions to the right feel good, and that's why the average goes down as variance goes up. Those are the unconditioned expected fitness values. But I also want to pay attention to a conditioned kind of fitness. I want to draw attention to the expected fitness as a function of noise, assuming that the realized phenotype is beneficial. Now, this may seem sort of arbitrary. Why should we be allowed to focus on things that are beneficial? We're just anticipating what natural selection is going to do. Natural selection is very good at finding beneficial variants, even if they're rare. So it's not the claim that higher noise organisms have higher fitness. No, on average, they have lower fitness, but they also have the potential to produce anomalously high fitness organisms. You can see that in comparing the marigold and blue distributions. And it turns out, these are analytic results, it turns out that as noise goes up, the expected fitness conditioned on a beneficial phenotype goes up with noise. Okay. So here are those results. Now what I've done is to throw away the full distribution. I'm only showing you two expectations as a function of sigma on the x-axis. The y-axis is the expected Malthusian fitness. The purple curve is the unconditioned fitness. As noise goes up, fitness goes down. That's Jensen's inequality. The marigold is the fitness conditioned on realizing a beneficial phenotype. As noise goes up, the expected fitness conditioned on a beneficial phenotype goes on. Okay. Now, this is just a toy model, but it actually has two features that I think are going to turn out to be biologically rather generic. The first is that the expected fitness across all noise goes down, and it goes down faster and faster and faster as noise goes up. That, in my mind, is a consequence of the fact that organisms are already pretty well adapting. And so if I just start throwing in more and more and more noise, things are going to get worse and worse and worse. The other feature that I think is important, and I predict will be generic, is that the condition fitness function goes up, but it goes up ever more slowly. And that's, again, in my view, a consequence of the fact that the populations are fairly well adapted. They're already pretty close to the fitness optimum. And that means that more and more noise stops paying. There's a sort of diminishing returns that derives from more and more noise, because now the population's already sampling phenotypes that are near or at the maximum. More noise stops helping. Okay, and so the fact that the purple curve goes down faster and faster and faster, its second derivative is negative, but the marigold curve goes up slower and slower and slower, its second derivative is positive, suggests to me, and these are analytic results for that toy model, but I want to argue that these are generic, these are going to be generic results, suggests to me that there will be some kind of equilibrium level of noise. If noise is very low, then a little bit more noise may be beneficial because it allows the population to take advantage of opportunities conditioned on being beneficial. But eventually, because this curve is getting slower and slower and slower, and this curve is going faster and faster and faster, there'll be a limit beyond which more noise is deleterious, because now the incremental benefits due to a fairly flat marigold curve are not sufficient to offset the cost of the ever steeper purple curve. Okay, so I've presented a toy model with analytic results that I think motivates us to think about there being some kind of equilibrium amount of noise in populations, some kind of selective equilibrium amount of noise in populations. But so far, we've only talked about the fitness effect of a new noise allele. We haven't yet talked about the population genetics of how does this thing get to fixation. So to get there, I want to review the kind of the grandfather, perhaps, of all alleles that influence noise, alleles that influence mutation rates. This work goes back to an experiment that Rich Zielinski started in the 80s, although high mutation rate alleles were described already in the 50s. So what Rich did was to propagate bacterial populations. And he's been doing this, and he and his acolytes have been doing this now for 75,000 or so bacterial generations. Every day there's 100-fold dilution. That gives rise to demography like this. Every day there are about 6.6 generations. It's been going on since 1986 or 9 or something like that. And so what's happened? Well, the first thing that happened, not surprisingly, is fitness went up. Because these populations are being grown in something called glucose-limited media. It's not the medium that the bacteria were living in before the experiment began, so there were opportunities for adaptation. But I want you to notice that fitness goes up quickly at the beginning. X-axis is time into this experiment. Y-axis is fitness relative to the founder. Fitness goes up quickly at the beginning and then slows down. That'll be important in a minute. What else happened? Mutation rates went up. So this is from a paper by my colleague and friend Paul Snikowski when he was a postdoc with Rich. And what Paul noticed is that so Rich had started 12 replicate populations. Those populations are arrayed along the x-axis. Don't worry about the nomenclature. The point to notice is that 3 of these populations have elevated mutation rates by 2 different assays. Mutation rate seems to have gone up between 10 and 100-fold. More recently, Ben Good, when he was with Michael Desai, sequenced 1,500 clones from these same populations across all 12 replicate lineages over 60,000 generations. And one of the first things he did was to look at the sequence data and simply count how many mutations had accumulated in each lineage at each time point. And that's plotted here. So the x-axis is time, the time from which the sample was drawn. Y-axis is the number of mutations relative to the founder. So the slope of these lines is some kind of proxy for the mutation rate. And what you can see is that 6 of the 12 lines have acquired a super accelerated rate of mutation. These same data, but now with a different dynamic range, a much larger dynamic range on the y-axis, We can't even see the 6 populations whose mutation rate is pretty low. But of the other 6, what I want you to notice is that the slopes in almost every case slow down again. The slopes return to a lower value, indicating that the mutation rate evolved to something lower after having evolved to something higher. So this is an example of a high noise allele arising and fixing in some populations, followed by a low noise allele arising and fixing in some of these populations. What happened? How do we understand these population genetics? The answer is indirect selection. So this is a schematic picture from a review that Paul Snagowski wrote. Each row is a generation, each horizontal line is a genome. And what you can see is that in the top row, the first generation, one individual happened to acquire a mutation that increased its mutation rate. And sometime later, an offspring of that individual acquired a beneficial mutation shown by the green dot or green box. I don't think I said it, but these were asexual populations. There's no recombination, which means there's no opportunity for the mutation rate allele, the red dot, to be separated from the beneficial mutation, the green dot. And natural selection does what it always does. It finds green dots and enriches for them in the population. So as time goes forward, as we move down the figure, you see that the green box spreads, and with it spreads the mutation rate allele. The beneficial mutation drives the mutator to fixation by a process called genetic hitchhiking. That's a consequence of this perfect genetic linkage that follows from the fact that these were asexual populations. Or in slightly different language, direct selection acting on the beneficial mutation drove the mutator to fixation by this process that we call indirect selection, sometimes called second order selection. That's why mutation rate went up. The exact same mechanism explains why mutation rate went back down, because now we look at the red dot as an anti-mutator allele, a new allele that has a lower mutation rate. And the consequence is that its offspring are not burdened by as many deleterious mutations as are the competing lineages that still have the high mutation rate. After all, most mutations are deleterious. We've said that already. Okay. What about in sexual populations? What about in circumstances where the phenotype is a developmental phenotype? It's not even encoded directly in the genome. Does indirect selection apply here? The answer is yes. If phenotype is resampled, however, if phenotype is resampled every generation, all natural selection can see are phenotypes. It can't see these modifier alleles. It can only see the consequences of these modifier alleles. So if phenotypes are resampled every generation, the natural selection can't tell the difference between the two alleles. And indeed, John Gillespie showed that high noise alleles in this circumstance, holding the mean phenotype constant, if phenotype is resampled every generation, high noise alleles are at an evolutionary disadvantage, because while they're rare, they're more likely to sample really crummy phenotypes, which causes them to be swept out of the population. This is really the central fulcrum of all stochastic population genetics, in my view, is what happens to a thing while it's rare. While it's rare, we cannot take any deterministic approximation, because if the thing is lost, it's game over. Once the thing, be it an epidemic in a population or a beneficial mutation in a population, or any other thing that you want to think about where your stochastic population model is appropriate, once the thing is established in the population, our deterministic approximations are pretty good. But at the beginning, not so. And this is the fulcrum of John's results. But it can be generalized. There is a space between perfect statistical association, permanent statistical association that we see in the asexual mutation rate modifiers, and absolutely resampling every single generation. And this problem was explored by Ioana Karja when she was with Josh Plotkin. So her model was, imagine a competition between a population that's carrying capital A alleles, in which the phenotype of the parent is transmitted faithfully to the offspring every generation. And now introduce a single copy of a little a allele, which similarly transmits the parent's phenotype faithfully with probability P, but with probability 1 minus P, it transmits a new phenotype drawn from, say, this distribution. What's the probability that such a little a, or in our language, high noise allele will succeed in the population? That's going to be shown here. The x-axis here is the memory parameter P, the probability that the parent and the offspring have the same phenotype, and the y-axis is the probability that the little a allele fixes in the population. This is a stochastic simulation of a single copy of the little a allele, which means that the relevant benchmark, if the little a allele were neutral, it would fix with probability 1 minus n. That's what that horizontal dashed line is. 1 over n. What did I say?
1 minus n.
1 minus n. No, the multiplicative equivalent. The reciprocal of n. Thank you. So here are Alana's results. These are simulation results, although she's got some very satisfying analytic approximations. As the memory parameter goes up, as the probability that the little a allele's offspring inherit the same phenotype as the parent, be it a good phenotype or a bad phenotype, as the memory parameter goes up, the probability of success of the little a high nose allele goes up. And it also goes up as we increase the width of the distribution from which phenotypes are drawn. So this fills in the space between the every generation I resample phenotype, where the high noise allele can never succeed, to the asexual mutation rate modifier, where every generation the phenotype is faithfully inherited. So I suggested that there will be, I haven't yet found it, I suggest there will be equilibrium amount of noise. That's motivated by this figure I showed you before. What are some of the factors that will influence the amount of phenotypic, the equilibrium amount of noise? The first is the phenotypic distance to the optimum. So the solid lines you've already seen, the dashed lines are redrawn, but now the phenotype of the parent is -1 instead of -2. It's closer to the peak, which means the opportunities for beneficial phenotypes are reduced. That's why the yellow dashed line is shallower and why the purple dashed line is steeper. So the closer I am to the fitness optimum, the less noise I want. In the limiting case, if I'm at the optimum, I don't want any noise. The modifier literature calls that the reduction principle. Or equivalently, because environments aren't changing, the distance between a population and its phenotypic optimum is some kind of quasi-equilibrium thing where the phenotype, the optimum phenotype is changing because the environment is changing, the competitors are changing, the pathogens are changing, the climate is changing. So the population is chasing that peak, some kind of quasi-equilibrium distance. The faster the environment is changing, the further the equilibrium distance to the peak is, the more beneficial noise is. That's one factor that will influence the equilibrium. Persistence time between the modifier allele and its induced phenotype. I just showed you that. Indeed, Oren Ecologie in Israel showed me a paper of his in which he showed that even if phenotypes are resampled every generation, if selection is stricter than fitness proportional, for example, if there's a truncation effect, in that case, even sampling phenotypes every generation can allow the high noise allele to fix. So those are important quantities that need to be considered. The population size matters. So this is famous work from Mike Lynch on the drift barrier hypothesis. Mike notices that the mutation rate shown on the y-axis is inversely related to the effective population size across life. This is from 2010. Now we're 15 years later. There are 200 points on this plot, but the regression is unchanged. And his explanation is shown in the lower panel. In his mind, natural selection is in the business of lowering mutation rate because almost all mutations are deleterious. In large populations, the pain of these deleterious mutations is more acutely felt, and that's why the mutation rate evolves to a lower equilibrium. That's for sure true. Student of mine, Eugene Rains, found another really interesting effect of population size. And when we read the literature, we discovered that Alex Whitlock in Christina Birch's lab had the same results. So our results are shown in the bottom. Christina's group's results are shown in the top. What are these figures showing? Population size is on the x-axis. The probability of fixation of the y-axis. The horizontal dashed line is the probability of fixation for a neutral mutation. So in fact, I've normalized the y-axis so that regardless of population size, the probability of fixation for the neutral mutation is 1. And what you can see is that in both panels, the high noise allele, the sexual allele in the upper panel, the mutator allele in the lower panel, Those are disfavored in small populations, but eventually become favored in large populations. And the low noise allele, the asexual organism in the upper panel, the low mutation rate, the anti-mutator allele in the lower panel are favored in small populations and disfavored in large populations. So there are lots of things here that need to be considered as we try to move to more quantitative, to any kind of quantitative understanding of where the equilibrium amount of noise lives. Okay, how am I on time? Okay, good. So I want to talk about implications of this work for two well-established domains of inquiry, because I think that, in fact, this framework subsumes these previous domains of inquiry. Modifier theory, I've already alluded to. Modifier theory was first, in some sense, introduced by R.A. Fisher. This is a quote from a talk he gave at the 6th International Congress of Genetics in Ithaca, New York, which is also the conference in which Sewell Wright introduced the fitness landscape. So an exciting meeting. Fisher writes, population genetic contributions are of two kinds, somewhat sharply contrasted, as illustrated by Haldane's subject, which was, can evolution be explained in terms of present known genetic causes? So that's kind of plain vanilla straight ahead population genetics. With my own address, which might equally well have been entitled, can genetical phenomena be explained in terms of known evolutionary causes? So you may or may not know that R.A. Fisher was really interested in the question of whether natural selection explains why deleterious mutations are usually recessive in humans. And he built an apparatus that invoked indirect, what we would call indirect selection, and here he is articulating the distinction between direct selection in the first and indirect selection. The latter, much more recently, but still many years ago, Carlin and McGregor tried to generalize some of these ideas. By this point, people have thought about dominance, mutation rate, recombination, assortative mating, migration, and they proposed that genotypes at what they called secondary or modifier loci alter the determination of parameter values, delimiting the genetical system, and mechanisms corresponding to the primary locus. So there's a lot there. What survived in general is this idea of alleles that influence parameter values like the mutation, like the recombination. And then Lee Altenberg, in his PhD thesis with Mark Feldman, defined modifier theory as the population genetics of loci that influence the transmission process. So to my mind, this is equivalent to what we call modifiers of reproduction. But I want to argue that the exact same theoretical apparatus applies to modifiers of noise during reproduction and modifiers of noise during development. So that's why I propose that modifier theory be generalized as in terms of the theory that I'm trying to develop here. Okay. Although modifier theory has largely been concerned with noise during reproduction, theory on the evolution of evolvability has been largely concerned with noise during development. And I want to kind of pull that domain of inquiry into the framework I've presented as well. So evolvability is an evolving population's capacity to generate heritably adaptive phenotypic variants. It matters that so for example, the evolution of pleiotropy is to me a really gorgeous example of this. If it turns out over long eons of time that natural selection favors simultaneously a high trait value, high values in two traits or low values in two traits, high for 10,000 generations in both traits, low for 10,000 generations in both traits, then there would be natural selection for mutations that tie those two phenotypes together. It increases the mutational target to move them if they always move in lockstep. And Richard Watson at the University of Southampton has done a lot of work thinking about how the genetic architecture of an organism encodes some features, statistical features of the environment that lineage has experienced over the last many eons. And so the evolution of pleiotropy is a perfect example. It allows populations to more reliably produce adaptive phenotypic variants. So the question then becomes, how and when will natural selection influence a population's evolvability? And this literature has been attacked for decades because it sometimes feels like there's a kind of teleological purpose that's built into these models. Somehow it needs to be that natural selection or the genome knows that in the future these two traits will be co-selected up or co-selected down. But that's not actually what's going on. It's only that it's reflecting past history that also had that statistical feature. And so I want to argue that, well, first I'm just going to restate what I just said. Focus on a population's capacity to generate adaptive phenotypic variation has, for many authors, suggested a kind of teleological paradox in all of this. And I want to argue that that's slightly missing the point because noise is a double-edged sword. Mostly noise is bad, but noise is also the capacity to generate adaptive phenotypic variation. Noise simultaneously, more noise simultaneously increases the capacity to generate adaptive phenotypic variation and deleterious phenotypic variation. I already introduced you to the idea of there being some kind of equilibrium, and I just want to say it again. Wherever the equilibrium lies in your favorite population, if the amount of phenotypic noise is too low, there will be natural selection to increase the amount of noise. This is modulo the persistence time, the population size, the model of selection, all the things we've already enumerated. But there will be natural selection, indirect selection, favoring high noise alleles, which you would look at and recognize as the evolution of evolvability. On the other hand, if the amount of noise in the population is too high, given all these other parameters, there will be natural selection in favor of low noise alleles because the cost of the noise is not being offset by the benefit of the noise. We might call that the evolution of anti-evolvability. Do what you want with that. But to me, it neutralizes the concern that the evolution of evolvability requires somehow comes for free. It doesn't come for free. Too much evolvability is a bad thing, if you think about it in these terms. Okay, so that's the main part of the talk. If there's still a few minutes, I'll just briefly talk about what we're doing now or what we'd like to do next. What we're doing now is trying to agree on a manuscript to upload to the bioarchive. There's some lovely theoretical work that I would love to find a student to engage in. I would like to quantitatively locate this equilibrium that I've alluded to twice as a function of these four quantities: the pace of environmental change, the persistence of the induced phenotype, the model of selection, and population size. I think that can be done using my toy model. That can be done rather easily in simulations, and I wouldn't be surprised, given the simplicity of the model, if some satisfying analytic approximations won't fall out. I also think it's important to incorporate mutations at the modifier locus. So Michael Desai wrote a nice paper when he was, I think, still a grad student, in which he pointed out that there will be some equilibrium level of mutation rate. But if you allow mutations at the mutation rate locus, at the modifier locus, there will be excursions in the population away from that equilibrium. Sometimes a high noise allele will appear and it will be carried to high frequency, even to fixation by a process that we've already seen, this indirect selection process. Now the population's mutation rate is too high, but we have to wait a little while for the anti-mutator allele to appear. The point of this theory is to develop a quantitative framework with which to evaluate data that I'll describe in a minute. So we need models that capture the most essential features of the biology. Finally, I've this simple toy model for what the fitness effects of biological noise look like, this normal distribution of noise and a quadratic fitness function. But there are lots of other ways of skinning that cat. And if you have a new model that you like better, you can just plug it into this thing and into this thing and see whether it really matters what model you use. So that's sort of on the theoretical side. On the experimental side, there's a sequence of things that need to be done. The first thing you need to do is demonstrate the existence of a modifier allele acting along some developmental or reproductive process. And the way you demonstrate the existence of such an allele is to do experiments like this. So if you think you have an allele that increases developmental noise, then you simply allow a large number of identical organisms to develop to adulthood, and you engineer or otherwise have on hand an organism that's genetically identical to this one, except it has the alternate allele, and you let an ensemble of genetically identical offspring on the right develop to adulthood, and you just compare the amount of variation in phenotype. Or if you're studying some modifier of reproduction, you have some single individual that you think has a putative high noise allele, otherwise genetically identical low noise competitor, and you allow this thing to reproduce 1 billion times and measure the distribution of inheritances. These inheritances are genetic, but also epigenetic, cytoplasmic, microbiome, anything else this is probably where niche construction lives. All the social things, a beautiful paper from Sally Otto last week in PNAS on how to connect cultural evolution with population genetics, and that's where this lives. And you demonstrate that there is heritable control, the amount of noise and inheritance. Once you've done that, you have to remember the first step always in making the case for natural selection is to measure the mutational bias. And so in other words, natural selection maybe pushes the population, even if there's no selection, the equilibrium amount of noise will be somewhere reflecting the mutational bias at that locus. So, and this polarity may vary with case. So for example, it's plausible that mutational bias favors increased mutation rate. It's easier to break a DNA polymerase than to make it more precise. But the evolution of asexuals, the appearance of asexuals from sexuals is probably genetically easier, genetically mutationally more likely than the appearance of sexuals from asexuals. What happens next is an important question that Christophe Haag and Thomas Lenormand and their group are working on. The next thing to do is think about indirect selection, think about direct selection. So the amount of noise in a system the amount of noise will have consequences for the energetic balance in the organism. So reducing noise can be costly, but other times increasing noise can be energetically costly. So for example, these same two examples, it's plausible that lowering mutation rate is energetically costly because the better DNA polymerase needs more ATPs to do the proofreading, but asexual reproduction is surely energetically less costly than asexual reproduction. You have to control for these things and also for other correlated consequences of noise. So for example, one of my favorite examples, in viruses, you can show that higher mutation rate polymerases shorten the generation time of the virus. That's just straight up beneficial. No matter what happens to no matter what the genetics of those offspring are, there's just more of them. So you need to think about effects like that. And once you've controlled for all of that, and by the way, there are a couple of nice reviews here that help us understand what these pitfalls are of over-interpreting our data. But then we can finally imagine asserting the action of indirect selection acting on noise. This will require the models that I'm proposing to develop, compare the amount of noise I see in some natural population among some set of alleles, or even just in a wild population, controlling for mode of selection, population size, rate of environmental change, blah, 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 blah. And make your case for indirect selection influencing the amount of noise. Okay. So that's it. Thank you for your attention. I want to acknowledge Brown University, which allows my wife and me to come to France every year with a little bit of financial support from these pots of money. I want to greatly acknowledge the CNRS, the CEF, the University of Montpellier, the EFD, and particularly Luis's team for taking in an orphan when Nacho's sabbatical in Portugal happened to fall in the same semester as my sabbatical here. And I'll just leave you with a plug. This is what I did during my sabbatical in 2021 when I sat in a beautiful apartment on the Jeu de Paume in the Écusson, is to start writing this textbook. If you're interested, you can scan the QR code, you can get a free sample, blah, 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 blah. I find it extremely useful in my own classroom, and I'd be happy to hear feedback from others as well. Thank you.
Thanks, Dan. So do we have questions in the room? Yes, there's one already. Okay.
Yes,
Yes.
thanks. So I was wondering during your talk how much this result depends on the exact shape of this quadratic phenotype fitness function, and you said that you will address that in future work as well. But do you have any ideas about how general this would be, like would a multi-peaked function have, or a multi-dimensional function have, or an asymmetrical function lead to loss of generality of these results, or what's your feeling about it?
Super question, thanks. My first reaction is if it's concave down, then the main result that more noise is on average bad, you'll survive.
Yeah.
One of my co-authors said, why do we make the fitness function normal too? I bet the math will be nice. And I said, yeah, but it stops being concave down and we get far enough out, so let's not do that.
You should.
But we probably should, exactly. And certainly if there's going to be a second peak, there has to be a zone of concave upness connecting the two. So I think it's extremely interesting. Yeah, I haven't given that any thought, but I really like that question. Yeah.
Pete?
I'd like to expand on that because we may think of the life in terms of adaptive landscapes and things like that. And if we think about people in physics, specialists of physics, who look at biology, one of the big properties of biology is that we start with a sum of stochastic processes and we should end up with a normal distribution. Well, that is what happens in physics. But in biology, we end up with separate peaks. So maybe if we have an adaptive landscape, you could select for noise that would be very much limited noise within one genotype, and then some very big noise. So a saddle shape with a small peak at the end.
Yeah. No, I think there's a lot there. At least for the reason I see why that's interesting is if there are going to be two peaks, there has to be a part of the so let's just stipulate that the phenotype remains one-dimensional for the moment. If there's going to be a second peak, then there has to be a region. If it's continuous, there has to be a region where it's concave up, and that directly favors high noise. So you could imagine a population that starts to feel such a valley in which high noise alleles are not, it's not even about the occasional beneficial consequence. It's just straight up good to be noisy because now more noise allows you to experience what's on the other side of the valley that could pull the population over. But I think it's extremely interesting. Like that. Exactly. Yeah.
Yanis, yes?
Actually, this is more of a comment than question, but I think the effect of a phenotypic variation on the probability of peak shift has been addressed by Mike Woodlock in a paper that has VIP. P stands for phenotype. I can't remember. Sometimes in the 90s.
And I'm sorry,
It could be in the 90s. And Mike Woodlock could be,
could be relevant. Yeah.
I had actually a question, which is unrelated to this. In your explanation for, or rather interpretation of the increase of, the decrease of fitness as of average fitness as noise increases, you interpreted this as something due to the fact that most genotypes were already adapted to their environment, and I actually failed to see where this specific assumption comes in the calculation. I somehow had the impression that it was a sort of general mathematical result.
Yeah, well, I think in the mathematics, it's a consequence of the fact that fitness goes down without limit as phenotype gets further and further from the optimum. So I think that's where it must emerge in the algebra that yielded those two curves, one of which drops faster and faster and faster, and the other one rises but slower and slower and slower. That's my intuition. You do not appear to be convinced.
I haven't thought about it.
Okay, so that's my intuition. And so as I was writing away and thinking about it, I thought, well, but that feels to me like a thing that's maybe not generically true, but very often true, no matter what the mapping from phenotype to fitness looks like, at least modulo a single feature state. And sort of concordant with the throwaway remarks that we often hear and make, that it's easier to break things in biology than it is to improve them. But it perhaps deserves some deeper thought. Thank you.
Yeah, I was wondering, don't you think we have a lot of data in the quantitative genetics literature that is not used because we put everything in, which is noise in the noise? And maybe if we have analyzed available data with your view, we can better understand the genetics of noise.
Yeah. I don't know enough, indeed, I don't know terribly much at all about quantitative genetic data, period. I was a computer engineer until 25 years ago, and so what I'm more exposed to are very recent whiz-bang single-celled visualization techniques and things like that that give us access to, that actually allow us to measure these distributions. And certainly there are variance terms in every roughly speaking, every single quantitative genetic treatment, but I would be really happy if you had some expertise to speak with you and try to understand whether there's something there to be pulled on. Yeah.
But in your perspective, you suggested that it could be interesting to do the quantitative genetics of noise. And I thought maybe you thought about it already.
So one point that comes to mind is that I recently read a paper, probably in yeast, that showed that developmental noise has a complex genetic architecture. They found, 4 EQTLs that interact to determine the expression level in some, I don't know the details of it. So that's a criticism of my model and my framing that's orthogonal to the question of, is phenotype one-dimensional or multidimensional? It undermines the claim that there will be single alleles that influence phenotypic noise along one axis or 2 axes or 7 axes. It's not surprisingly, it's surely going to be much more complicated than that. And so maybe one reflection of your question is, what would a quantitative genetics approach look like, as opposed to an approach that assumes they're just single loci that are controlling this noise? I don't know the answer.
I think there is some data towards gene expression analysis where people perform quantitative genetics things on these types of traits, and you have one trait per gene, and then they can see if the residual variance of the trait varies in the types of things. I have one question that came to mind when you were talking at the end about empirically simulating that. I was wondering Since the magnitude of noise seemed somewhat as important as the memory in some of this process, I wonder if there's to be some correlations between that empirically, that some mechanism that produce more noise, they produce also more or less memory, and these things might actually be correlated. Depends on, I don't know, regulation of epigenetic regulation of expression and things like that. I think there's something along these lines that might be interesting.
Yeah, that's really interesting.
We don't know yet there.
Yeah.
Okay. Any questions online? No?
No.
Then I think we can leave it there and thank Dan again.